So, to continue where I left off, I need to cut out that last like 30 seconds. So you're going to make 3-phosphoglycerate. Alright, so this glyceraldehyde 3,1,3-bisphosphate, this phosphate is going to become 3-PG, which is 3-phosphoglycerate. Alright, instead of glyceraldehyde 3-phosphate, it's 3-phosphoglycerate. Okay, 3-phosphoglycerate. Which is then going to isomerize. It's going to make 2-phosphoglycerate, so 2-PG. Also not a very important step. Not really that well regulated or anything. And then your 2-phosphoglycerate is going to become phosphoenol, so it's called PEP. Right? PEP is super important. It's high energy. It's used in anaerobic respiration. If your body gets desperate, your brain and your red blood cells might use it. This is called phosphoenol pyruvate. You notice we have the pyruvate up here, and we have the pyruvate down here. They're super important because this is what we're trying to produce. So now what happens is you're going to take your phosphoenol pyruvate, and this is your last regulated step, and you're going to use what's called pyruvate kinase. So we're going to take this pyruvate, and I'm going to add kinase on here, and you're going to make pyruvate. Alright, so, and notice this had a phospho. This had 2 phospho groups. This glyceraldehyde had 1,3-bisphosphate. So what happens is when you lose this first phosphate group, you produce ATP. And because there's 2 of them, you actually produce 2 ATP. And then, down here, when you convert phosphoenol pyruvate into pyruvate, you also lose another phosphate group, which means you make 2 more ATPs. Okay, so in our energy investment stage, you spent 2 ATPs to make fructose 1,6-bisphosphate. But in the end, you pay off, you actually make 4 ATPs, which means you get a net of 2 ATPs. Plus, there's these NADHs that I talked about earlier. Those are electron and high energy carriers that are used in the tricarboxylic acid or the Krebs cycle. You also made 2 of them. So I'll show you the bottom here where you make pyruvate. Okay, so let's talk about this. There's 10 steps. 3 of them are regulated. The first one, where you convert glucose into glucose 6-phosphate. That is actually regulated by the amount of glucose and by the amount of glucose 6-phosphate in the cytosol. So inside the cellular matrix of whatever random cell you are, if you have too much glucose 6-phosphate, the enzyme hexokinase stops working because there's just too much glucose 6-phosphate, which makes sense because then the cell doesn't need that glucose, so it sends it back into the blood, and then it goes to other cells that might need it, or it goes to the liver for storage. Or if your body can get rid of this glucose 6-phosphate, you can make glycogen, which you can use to store it for later. But sometimes your muscles and your liver, share the duties for that, whatever's most efficient for your body biochemically. And I'm going to talk about insulin and glucagon later because they regulate all this. All right, and then we got to talk about this phosphofructokinase 1. Okay, so phosphofructokinase 1 is super important because that's the second ATP investment step, and it's really heavily regulated because once you reach this step, you make fructose 1,6-bisphosphate, you have to do glycolysis. All right, you can't really stop halfway through without finishing and getting the energy and going to the TCA cycle. So the body doesn't want to do it unless it has to. So phosphofructokinase 1 is regulated by something else called, so this is fructose 1,6-bisphosphate. When you have a lot of fructose 6-phosphate, your body uses a different enzyme and it makes fructose 2,6-BP. Fructose 2,6-bisphosphate. Okay? Now fructose 2,6-bisphosphate activates phosphofructokinase 1 and says, hey, you need to phosphorylate. We got a lot of energy. We got a lot of glycolysis that needs to happen in the body. It just needs to go on. Other things that tend to activate phosphofructokinase 1 would be AMP, ADP, because it says your body, hey, we need energy. Let's do some glycolysis and make some energy. Things that would inhibit it would be ATP, okay? Or just a lack of fructose 2,6-bisphosphate. Another thing that can affect it is citrate, which comes out of the TCA cycle. So citrate, if there's a lot of TCA and a lot of energy being going on, the citrate leaves and it inhibits this guy. The last one is this pyruvate kinase. Okay, the last step, number 10, is the other really regulated step. And pyruvate kinase is regulated by ATP, understandably. It's inhibited by ATP. It's also inhibited by parts of the TCA cycle that says, hey, we got a lot of energy, we don't need this to happen right now. But it's also activated by fructose 1,6-bisphosphate. And this is what's called a feed-forward activation, where the enzyme from the earlier step goes to the later step to activate glycolysis, so that, because you don't want to stop halfway through here, then you have a bunch of, these are actually really high energy intermediate molecules, and you don't want to have a lot of high energy sitting around in the cytosol unless you're going to put it somewhere. You don't want high energy. It's the same reason you just don't want a bunch of energy sitting around. You know, do you want a pile of gasoline under your bed at night? No, you're going to store it in the basement or somewhere where it's more likely to be useful, which in this case, you know, that's why we have glycogen in your body. Okay, so there's 3 steps that are really inhibited by glycolysis. So please comment underneath this. Let me know some things I could do to make better. I definitely, this board's a mess right now. Also need to make sure I remember this off the top of my head. I'm trying to do this to study as well as teach. So if you have any tips, or if there's questions you have, or if you want to ask me questions and try and stop me, go for it. Thank you for your time. Have a good day, guys.